processes or the steps of actually oxidizing this glucose and yielding the ATP. So the three processes involved are glycolysis, the Krebs cycle, and the electron transport chain, also called the ETC. And so glycolysis and the Krebs cycle occur via substrate level phosphorylation. So this is where we take a phosphate from another molecule, if you remember that example from a few slides back. Whereas the electron transport chain is basically using an inorganic phosphate or free floating phosphate and generating ATP that way. I'll point out some important differences here again. Glycolysis happens in the cytosol of a cell, whereas the Krebs cycle and the electron transport chain occur in the mitochondrial matrix. We'll talk more about the details. Also want to point out that substrate level phosphorylation can happen in the absence of oxygen, whereas oxidative phosphorylation must have oxygen. So looking at the steps here of how glycolysis occurs, this is basically 10 enzymatic reactions that occur sequentially. And we break down one molecule of glucose to yield 2 pyruvate molecules. So halfway through these enzymatic reactions, the reaction occurs twice, so we yield 2 for every single glucose that we invest. Now let's look at what we are investing. So we invest 2 ATPs. We also invest 2 NAD+. Now NAD+ is that carrier molecule we talked about when we looked at nucleotides. And in its oxidized form, it is NAD+, meaning that it's not carrying electrons. But in its reduced form, we add 2 electrons onto that carrier, and it's going to be NADH plus 2H. So those 2 electrons will kind of hitch a ride when this NAD gets reduced. Again, this occurs in the cytosol, and oxygen is not consumed, nor is carbon dioxide produced. So let's look at the overall reaction equation. At the bottom here, we have glucose, 1 molecule. We invest 2 NAD+ in the oxidized form. We invest 2 ADP and 2 phosphates we're going to take from other molecules. And that yields 2 pyruvate, 2 NADH+, and then 2 ATP. Now the 2 ATP is net ATP because it actually generates 4 ATP. But because we had to invest 2 ATPs, then it's only yielding us 2 net ATPs. Okay. Let's look more closely at this here. So there are 2 phases involved. There's the energy investment phase, where we invested those ATP. And then at the bottom here is the energy harvesting phase, where we yield 4 ATP. Remember, this happens twice. From aldolase onward, this reaction happens twice. So we yield 4 ATP for the 2 ATP that we had to invest. And again, you don't have to memorize these molecules, these enzymes. If you go on to grad school, you probably will, or if you decide to do research in this area. But basically, you just really need to understand what's invested, what's yielded, and how many times this reaction occurs and where it occurs. So this is another slide showing the reaction here. Now, from glycolysis, we have our 2 pyruvate, and that is shuttled off to the mitochondrial matrix, where we have this small linking step. So this linking step converts pyruvate to another important molecule, acetyl-CoA, which can go on to the Krebs cycle. So in this very short reaction, pyruvate is entered. Remember, we have 2 pyruvate that we yield. Pyruvate is entered here. With the help of the enzyme pyruvate dehydrogenase and coenzyme A and acetyl, we generate 1 acetyl-CoA per pyruvate molecule invested. We also release NADH in the reduced form. So we're going to hitch a ride for 2 more electrons, and we're also going to release carbon dioxide as a byproduct. So all of the reduced electrons or the reduced NADH plus molecules that we're making are actually going to the mitochondria for oxidative phosphorylation. So let's jump back to this slide, where we made some NADH here as well. So as you can see, the NADH that we made here is heading to the mitochondria for oxidative phosphorylation. The NADH that we make here as well is also going to oxidative phosphorylation. Now, the Krebs cycle, also called the citric acid cycle or the TCA cycle, is another cyclic metabolic reaction or pathway that happens inside the mitochondria. And this is where we produce some more ATP. So 1 ATP per acetyl-CoA. Remember, we have 1 acetyl-CoA per pyruvate, and we have 2 pyruvate per glucose. Okay? So we generate 1 ATP here. We generate some more reduced NADH, some FADH, which is another form of that carrier, also carrying electrons, and then some more carbon dioxide as a byproduct. And again, these carriers are taking those electrons to the ETC. So basically, this cyclical process yields us those important factors: NADH carrying some electrons, FADH carrying electrons, and just a small amount of ATP. Okay, so far we have seen that we've yielded some ATP, right? Around 4 ATP molecules per molecule of glucose between glycolysis, the linking step, and the Krebs cycle, okay? So that's not a whole lot of ATP per glucose. So most of the ATP we'll see is actually made in this next step, oxidative phosphorylation, which happens via the electron transport chain. And this is basically where we move electrons down the electron transport chain, and we take that energy that is used as electrons are passed to yield ATP. Now this step must use oxygen. This is the whole reason that we breathe in oxygen, in order to be the final electron acceptor and to generate ATP by ATP synthase at the end. So the electron transport chain is a chain of molecules. So we have quite a few molecules sitting in the inner mitochondrial membrane. So if we remind ourselves of the structure of the mitochondria, there's an inner membrane, an outer membrane, and then there's a matrix. So in between the inner membrane and the matrix, we have the inner mitochondrial membrane, and this is where the electron transport chain occurs. And basically, we pass those electrons that we've been talking about, that were shuttled off to the mitochondria. We pass them from complex to complex, and we generate a gradient of hydrogen ions. And then that gradient facilitates the movement of those ions, which then yields our ATP. Okay.